speaker here. Let me share with all of you his curriculum vitae. Okay. So can you see my slide well? I'd like to introduce you to our next second speaker is Dr. Apotheker Muhammad Amrun Hidayat. So he is from the Pharmaceutical Biology Department of our Faculty of Pharmacy, University of Jember. And his last education is in 2022 in University of Erlangga as doctoral of pharmaceutical science. And his areas of expertise is pharmacognosy and also optical, chemical, and biosensor. And his latest publication already published in the 2022 about the paper-based sensor for some biomarkers. And he also awarded some numerous research grant as a reviewer also. And so without further ado, he will present about the very interesting topic also about the application of paper-based sensor on food, beverage, and medicinal plants extract determination. So this is also interesting in our medicinal plants knowledge. So let Mr. Amrun, can you hear me? So maybe
yes.
You can unmute. Okay, yes. So not waiting for too long, just begin the presentation and time is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. First of all, I would like to appreciate the Committee of International Conference of Medicinal Plants that give me the opportunity to share my research for so long. And without waiting again, please let me share my PowerPoint. Is everybody can see my PowerPoint?
Yes, very clear. Yes.
Okay. So today I will talk about application of paper-based sensors on food, beverage, and medicinal plant extract determination. First of all, we have to know what is a sensor. Sensor is a device that detects events or change in quantities and provide a corresponding output. We know that a mercury thermometer is an example of a physical sensor. It converts the measured temperature into expansion or contraction of a liquid, which can be visualized on the calibrated glass tube. Another example is thermocouple, which convert temperature to an output voltage that we can see in the display. So there are two types of a sensor. First of all, is a physical sensor. Physical sensor is a device that provides information about physical property. And for example, is pressure sensor, temperature sensors, etc. And secondly, is chemical sensor. It is a device that transforms chemical information, ranging from the concentration of specific sample component to total composition analysis into analytical signal. Example of the chemical sensors is carbon dioxide and pH and ion meters. So we go more deeply about chemical sensors. It is defined as an analytical device incorporating a chemical material or region that can recognize or react with chemical analytes, whether in the solution or even in the atmosphere, with a physical temperature. So transducer is maybe look like a detector in a chemical instrumentation. So in concise, chemical sensor consists of a region plus transducer. So what about the biosensor? Principally, biosensor is analog with chemical sensor. The difference is it is used as biological material as a biological or biorecognition. So it can recognize a biological or chemical analytes in any type of sample. So the example of a biomaterial or biorecognition is enzyme. Mostly the biosensor is developed by using enzyme and also antibody. So, and DNA, even whole microorganism such as E. coli can be used as a sensor or biosensor material. So in concise, a biosensor is made by a bioreceptor plus a transducer. So today I would like to share my expertise and my experience for developing paper-based sensor. So in historical view, a litmus paper is basically a paper-based sensor. It measures pH. So if we have acidic solution, it converts blue litmus, red litmus into blue color litmus, and so on in opposite. And then secondly, in the medicinal plant fields, we already know that chromatography paper. So basically a paper chromatography, which we can use as a spot test for identification of many compounds and phytochemicals of medicinal plant is basically is a paper-based sensor, which is a simple paper-based sensor. So why we use a paper? First of all, paper is a good medium of course for test because it is white, which provide a strong contrast with color test. Of course, white background is more desirable than a black background. Secondly, it has a high surface to volume ratio, which facilitates accelerated evaporation of solution. In this case, whether a region or bioregion results in a concentrate analytes or a region or bioregion on the sensing zone. Thirdly, it is extremely cheap and easy to handle. So everybody know papers, everybody work with papers. So everybody can make a paper-based sensor, actually. And it can be designed as a lateral flow, just like a pregnancy test or flow analysis that run by capillary action without external pumping, such as, for example, high pressure liquid chromatography and so on. And lastly, it is disposable and more eco-friendly than plastic-based cuvette or microwave plate. So here is an example of glucose sensor and protein sensor. It is to analyze glucose and protein in urine sample simultaneously. So if we ask what is the application of the paper-based sensor, so if we look at Indonesian herbal pharmacopoeia, there are several tests for phytochemicals of medicinal plant extract. Number one is marker compound detection. Let's say, for example, like quercetin or maybe sinensetin, Secondly, is total group of compounds, let's say as a total polyphenol content and also total flavonoid content. And number three is a TLC profile of a single extract or raw material, or as a simplicia. So can paper-based chemical sensors or biosensor apply to this chemical test according to pharmacobehavioral Indonesia? So in my research, I always use paper-based chemical sensors. I developed what is called a paper microzone plate as a select support. So it is firstly introduced by Professor Whiteside group. It is used as an alternative for plastic-based 96 microwave plate. So basically, it is a filter paper, which is screen printing by using rubber ink to make a circular pattern at both sides of the paper. So basically, for sensor development, the region or bioregion, which in case in the biosensor, are immobilized on each microzone as a sensing zone. And then we have a standard or sample solution is applied, is added to the dried region of bioregion. And then the color chain of the sensor is captured by a flatbed scanner here and further quantified by image processing program. For example, ImageJ, which is known this technique as a scanometric method. You can see what is scanometric method in my publication. So the first sensor I would like to share with you is single marker detection, or in this case, quercetin sensor. It is inspired by the work of Askal et al. In 1997. So if we have a flavonol, it can be analyzed spectrophotometrically using N-bromosuccinimide. Fortunately, this region is reacted selectively with quercetin, forming a deep violet color, which can be analyzed at 510 nanometers. Whether the other flavonols, such as camphorol, or other like luteolin, is not forming purple color with NBS. So what happened here, quercetin is reacted with NBS to form its bromo derivative, which has a purple color. So we adopted this chemical reaction into the paper-based form. So in a paper microzone plate, I immobilize this NBS region, and we mix the standard quercetin or sample extract with 1% phenol to stabilize the pink color, which is arise after the sensor is reacted with a standard or sample solution. This purplish red color can be quantified as the analytical signal. Here, this is a blank sensor, and this is a quercetin in a concentration ranging from 1 to 10 millimolar. So, secondly, if we look at WHO, there are tests that is recommended by WHO, such as heavy metal contamination, and what I also developed formaldehyde sensor. Basically, as a pepper chip for formaldehyde detection, it is actually a biosensor which use enzyme, alcohol oxidase, which can oxidize formaldehyde, and the oxidized formaldehyde further reacted with tetramethylbenzidine to form a blue-yellow color, which can visually detect it, or we can quantify it using imaging program. So after we conduct research, this chip can be applied for formaldehyde detection in noodle and meat samples. So I'm sorry, this is a wrong place maybe. The quercetin sensor uh, used for quercetin determination in guava leaf extract sample. We know that guava leaf extract is a starting material for jamu or herbal, standardized herbal medicine, and also phytopharmaca. So measuring quercetin is very important. And our research show that the developed sensor can result, the result of development sensor for quercetin determination is not quite different with the standard TLC densitometric method. Okay, and then the second application of pepper-based sensor is for total polyphenol content. In this case, I quote the Archeoli et al. 2013 for the developing a biosensor for total polyphenol content. It is used as tyrosinase enzyme and chromogen MBTH. This enzyme can oxidize the substrate, monophenol or orthodiphenol into its quinone. And the quinone compound can further react it with MBTH to form pink colored agar. So in their research, the method is developed for PPC determination of line sample, expresses as L-DOPA equivalent. Another example of tyrosinase and MBTH-based biosensor is a biosensor for determining many polyphenols such as 4-chlorophenol, catechol, M-cresol, and paracresol in the water samples. It is basically the same mechanism and use this very same bioregion and chromogenic region. Here, dopamine as used as L-DOPA is used for the biosensor characterization. So our publication used very same mechanism of biochemical reaction. We just use enzyme, but I'm sorry, we use catechin as a standard for validated sensor. Sorry, this should be catechin. So here, if we have a sample containing catechin or polyphenol, it can it can change the colorless biosensor into red color biosensors, and the color change can be recorded using a scanner, ordinary flatbed scanner, and the color intensity can be quantified using an imaging program. And this developed method is applied for green tea beverage So our research is different with the previous biosensor research that we use a catechin as a standard and to express the total polyphenol content. We know that catechin is a basic structure of green tea polyphenol. So it is more related in the for sample than the previous biosensor. We know that L-DOPA is not related with the wine sample in the previous research. Okay, the next application, we use a chemical sensor for total polyphenol content. Basically, it is used sodium pyriodate to generate catecholamine. Sodium pyriodate is an oxidator which is used as a oxidator to replace tyrosinase or to study kinetic of a tyrosinase enzyme for many polyphenols such as catechin, chlorogenic acid, etc., etc. And it is more stable than tyrosinase and the reaction is speeder than the tyrosinase. So we developed the sensor in the form of a street test, and the developed method is applied for the TPC determination of green tea beverage. And TPC is expressed as catechin equivalent in ppm. Here, the result show that our developed method, the result is quite similar with that spectrophotometric method. So the similar platform, the similar chemical mechanism is also applied for total polyphenol content in the coffee samples, in coffee beverage samples. But the difference is we use chlorogenic acid as a standard. We know that CGA or chlorogenic acid is the main polyphenol found in the coffee. So we use it as a standard, and we calculated the TPC in milligram per liter chlorogenic acid equivalent. So basically, the biomechanism or the biosensing mechanism is the same. CGA is oxidized by sodium pyriodate to form CGA quinone, and the CGA quinone can react it with MBTH to form the CGA quinone MBTH complex, which has a pink color at the sensor surface. So our result show that the developed biosensor can be applied to many coffee samples such as Arabica, Robusta, Greenland, Grown, and Mogus or Luwak coffee. Okay. The next application of pepper-based sensor is antioxidant capacity. We differentiate antioxidant activity with antioxidant capacity. If we tested the single compound, we call it antioxidant activity. But if we test the multi-compounds such as extract or fraction, we call it antioxidant capacity, or in other literature, often called total antioxidant capacity. Here, I show you the example of the micro pepper analytical device. It is fabricated by lamination method using ordinary plastic in plastic film, the plain plastic film, which is we made some hole and the filter paper inserted between two plastic, and it is laminated, and UPPH is immobilized on the filter paper and to use as a sensor. Here, the sensor is show a linear response toward gallic acid, and the sensor applied to green tea samples. So we also made PPH-based sensor, but instead using a laminated film, we use the similar paper microzone plate as our previous publication. So basically, it is made simply by immobilizing PPH into the paper microzone plate, and then the standard or sample solution is simply added to the sensing zone, and the total antioxidant capacity is determined or calculated as millimolar gallic acid equivalent. So this antioxidant sensor also applied to many plant medicinal extract such as West Indian elm leaf, red guava leaf, white guava leaf, stone breaker leaf, kutipling or green tea leaf, and this is a Jati Belanda. And then the next application of antioxidant sensor is it is based by cupric region. We use the same paper microzone plate, but we change the region. Here we use the cupric region, which consists of neocuproin, ammonium acetate, and cuprochloride. We immobilize the cupric region into sensing zone. And after that, the standard of sample solution is simply added to the sensing zone. And here we can see the blue copper reagent. If reacted with the antioxidant sample, it turns into yellow color. So the total antioxidant capacity calculated as millimolar routine equivalent. We also tested many medicinal plant extract such as turmeric rhizome or kunyit, guava leaf extract, whether red variant and white variant, and also black tea leaf and green tea leaf extract. And the results show that the developed method show no different observed between two methods statistically. And then maybe the last application of a paper-based sensor is a sensor array. Simply to say, sensor array is multiple sensor that contain different regions that used for simultaneous determination of many or different purpose. Here, at the Luktafon and Maka 2016, the total polyphenol and total flavonoid and also antioxidant capacity can be tested simultaneously by using paper-based microfluidic devices. Basically, it is choline Q and aluminum chloride and DPPH are individually immobilized onto the sensing zone here. And then after the standard or sample solution added to this sensing zone, the sample or standard solution is the sensor is dipped onto the surface of the solution, and then the solution is moving by capillary force, and then reacted with the region, each of region. So simultaneous determination of TPC, TFC, and antioxidant capacity calculated as milligram gallic acid equivalent, quercetin equivalent, and DHT equivalent. So here the calibration curve of total polyphenol, total flavonoid, and antioxidant capacity by using the sensor array. And then the next sensor array is a paper-based device with cross shape. It is fabricated by screen printed technology using polymer, which is a hydrophobic barrier. So the LBTS and cuprack and polycoccal region were deposited on the pre-treatment and the detection zone here. Basically, LBTS and cuprack and polycoccal, all of them is consisted of two regions. For example, LBTS, here, LBTS, they immobilize the LBTS and calcium and potassium persulfate here. And cuprack, it is the cupric chloride and ammonium acetate and neocuprine immobilized separately here, and the polycoccal and sodium carbonate also immobilized separately. So this sensor work that if we put sample solution or standard solution here, the fluid or the liquid is moving through all the detection zone by using capillary force, which facilitated simultaneous detection of antioxidant capacity by two methods. It's actually MBTS and the cuprack as antioxidant capacity and total polyphenol content simultaneously. So here the sensor array is applied to determine TPC and antioxidant capacity of a tea sample. For example, oolong tea, black tea, and red wine sample. And the last one is my research is called color profiling using antioxidant sensor array. Basically, it is consisted. The sensor array consisted of the three different regions: the cuprack, the IBTS, and polycoccal, which is immobilized independently and multiplied here 3 times, and different sample, for example, guava leaf red variant, or guava leaf white variant, and turmeric rhizome extract is added here. So after several times, actually in second to several minutes, the sensor array change the color. Here, every extract has a different color profile. So this is the actual color of the sensor array. This is before the sensor array reacted with the sample, and this is the color of sensor after reacted with different samples. If we analyze this their color intensity, which is red, green, blue, and average red, green, and blue, we put it into analysis, multivariate analysis. We have what we call a color different map. Color different map is a substitution. If we measure the color intensity of the reacted sensor array, subtracted with the blank sensor array, we have the color different map. And the color different map is further analyzed using HCA, hierarchical cluster analysis, and also principal component analysis. The results show that every each sample is clustered individually and clustered with their own type. So here, the GM sample or guava leaf red variant. This is the guava leaf white variant, and then this is a rhizome. Sorry. This is a turmeric rhizome, and this is a guava leaf red variant, and this is a guava leaf white variant. And if we look at the principal component analysis, we know that all the sample are placed or located differently in the PCA score. So the conclusion of what we have done so far is, firstly, paper-based sensor has a long historical use in chemical analysis, while its practical use is continuously expanding in chemical and pharmaceutical field. Secondly, the paper chemical sensor, along with scannometric techniques, can be applied for determining a single marker, TPC, TFC, and total antioxidant capacity in food, beverage, and medicinal plant extract. The developed paper-based sensor with the scannometric technique is simple, easy to operate, and more economical than the standard method, which is suited for the lab with limited resource and for the on-site analysis or field-safe analysis. Here the reference we have published in some international journals. Yeah, and then thank you everybody. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for very interesting presentation, and also I think paper-based sensor is simple yet promising. I think, yeah, for especially in the accelerating the finding of more potential medicinal plants in maybe, for example, in the forest, because we can imagine that you only have to bring your paper. Instead, you have to bring your spectrochromatography. So you just bring your paper and you can go everywhere in the world, and then you can find potential plants for our medicine. So this is very interesting topic, and maybe we can discuss in the discussion part. For the participants, please raise your hand if you want to ask to our speaker today, for Ms. Astuti and also Mr. Amrun. Yeah, let me see. So still there is no one. As we can see in the chat room, it's already a discussion there. But maybe we can also repeat it again lively. Let me read the first question for Miss Astuti from Mrs. Ari Nurhayati. Yeah, I think it is a very good work of you. I'm very much agree with your idea. However, now we need more of the coherence between political and grassroots movement to increase health, conserving the traditional heritage. However, one problem should also think is the environmental sustainability, including to conserve the soil itself. Maybe Ms. Astuti can repeat again your comments and also your insight regarding this idea.
Okay, thank you, Ms. Afifa, and thank you also to Miss Ari Nurhayati. Nice to see you and your question also, and all of the participants, of course. Yeah, I think once again, thank you very much for your appreciation. And then, yeah, what you said is already right, that yeah, it's challenging. It's never ending work, you know. So because we need to work with all of the section, right? As I already mentioned, that we need to make it balance between institutional level and then economical area, and then environmental and social culture. So it's really easy to say, but it's not really easy to do. I mean, to execute it, because, for example, in some cases, based even based on my review paper, I already tried to see. Sometimes it doesn't. How can I say it? Like there is a gap. Yeah, there is a gap between, for example, like when you try to apply the policy in the local level or even the regional level, it doesn't link to. I mean, it doesn't. How can I say it? Like we couldn't see the line between the policy which is applying which is applied, in the local level and national, or even in the global level. So this is a huge challenge. So we need to make it in balance, or even try to fulfill the gap. But my suggestion in this case, especially, we can start, especially in the regional level. It would be effective, because regional level can understand well about their area. For example, in our case here in East Java, so it means that the government in the East Java can coordinate or can encourage all of the stakeholders under the, what is that, like under the authority, and then each of the stakeholder can provide the like, let's say, like their opinion or even their insight, or how they can manage their resource based on the local specific level. Yeah, let's say the government of East Java can coordinate with Murubetiri National Park, for example. So it means that we can focus on, and then of course with the government of Jember, for example. So it means that if all of the stakeholder can work together on the same table, on the same page, also how to see what is the problem basically. So then we can also find the solution. Yeah, even though it is not really easy, but at least we can do it. So that's what I can, and especially what is it? I hope that it can answer. I mean, it can be the answer. Thank you.
Okay. Maybe Mrs. Ari want to reply to the comments. Okay, let just going to Mr. Amrun. There is also a question from Fajrin. How has the application of this sensor? How has the application of this sensor been in practice until now? Is it applicable if you want to make it in large numbers, such as in industry scale, for example? Maybe you can answer this question, Mr. Amrun.
Okay. Definitely, I already answered the question from to Ari. Yes, it is. Scanometric is basically is a colorimetric technique which measure the color intensity change after the sensor or biosensor is reacted with the sample or the standard solution. So the question from the Upajrin, how has the application of the sensor has been practiced until now? Frankly to say, our developed sensor, the stability is very low. Let me show you. Example of perhaps I want to share screen. Is everybody can see here?
Yes, can see.
Yes. Okay. Here the example of a biosensor which is used as a total polyphenol content of green tea sample. Here we know that from the figure 5, the stability is hold for only 9 days in freezer temperatures. Whether it is stored in a chiller temperature, it's only stand for 5 days. What about when it's stored in the room temperature? It is not lasting more than 1 day. So yes, I have to confess that the stability of the chemical, the pepper-based chemical sensor and biosensor is very limited. It has to be stored in the chiller or the freezer for the better performance. So in the industrial upscaling, I think I would like to say I don't dare to to scale up the sensor before we have to sure that their stability is very high. For example, as a pregnancy test, pregnancy test is actually they immobilize the antibody for the hormone human gonadotropin, which is stable for a couple months. But in our work, the stability is the major problem that we face here. So we cannot apply this the developed sensor. We have to make it recent apparatus, or we have to make it new, and we have to store maybe for the filter analysis we have to store it in a dry ice box, or we have to make the sensor new. So we separate the region of bioregion in the bottles, and we made the sensor immediately. And yes, but in a practical point of view, it is can be used a field trip study, which is I think maybe it can help Dr. Sri Astuti in the field trip to detect on the screen what plant contain flavonoid, what plant contain polyphenolic, etc., etc. By using a very cheap instrument, we have only used the ordinary office scanner or flatbed scanner. More recently, we also can use our smartphone and a free program, Android program, to measure color intensity. Almost every Android program which can measure color, we can use it as a interface to calculate any samples or any analytes indirectly. Thank you, Dr. Afifah.
Okay, thank you. Okay, it's also rise another challenge, I think, but maybe it can trigger future research for the next research, and hopefully we can have paper-based sensor that really stable, and then we can bring everywhere. Okay, there is still a question for Miss Astuti from Miss Puspitasari. I think you also already answer in the chat room, but because there is still no one raise their hand, maybe I just
Miss Afifah, I'm sorry, can I just interrupt a bit? I
yeah.
think I see Miss Ari Nurhayati say something there. Could you see that, please?
Okay. Please, Miss,
Still unmute, maybe,
can
or
you
something.
unmute yourself?
Yes, yes. Now
Okay,
I can. Yes. Okay.
can hear you.
Thank you. I'm asked to Mrs. Astuti. I would like you also, but and then how to make like you work in Germany, is it now? Is it?
Study, basically,
Yeah,
Miss
yeah.
Nurhayati, yes, study.
Yes, yeah,
Yes.
yeah. What I think is the difference between Germany and here is what I mean is for how to conserve the soil itself in Indonesia,
Okay.
because sometimes the difficulty is the physical soil itself. Some kind of people assault the soil, but actually the soil is good. Is no heavy metal contamination in the soil, and then after that they because they need the money and then they sell the money. So it is difficult for the for the how to make a sustainable of the soil, also to conservation of the soil. This the problem in Indonesia also. So,
Okay.
yeah.
Yeah. Yeah. So, can I just say something,
Yes.
Miss Nurhayati? Yeah. So, what you mean here is about the soil conservation, yeah. Especially you concerned about soil conservation, right?
Yes. Conservation.
Okay.
Yes.
Soil conservation and
And
as I
sustainability.
as I understood that people also try to sell the soil material. Is it what you mean? Does it
Yeah.
what you mean?
The
Or
soil itself, the land itself, the land
oh,
itself.
you mean the land?
Yeah, the
Yeah, the
land
land.
itself. Yeah, the
Okay.
land itself.
In the land use, yeah, in the land use
Yeah, the land use.
level. Okay. Yes. Yeah, it's yeah, it's a, once again, it's a big challenge. Yeah, it's a big
Big
challenge,
challenge.
Mr.
Yeah.
Hadi. Yeah. So I think but this is the very. I mean, I would like to underline one point, just only one point. It is related to the regional planning map, or even something related to how the people or even the government can be strict, yeah, can be strict to the land use management. For example, which area that the people couldn't sell it anymore, and then should be protected, for example, and should
Yes,
be
it's
classified
correct.
into some categorization, right? So,
Yeah.
I mean, this is the more important thing that the government should concern on. This or so should focus on in this area. I mean, yeah, because if the regulation couldn't handle this kind of situation, means that the people can just do anything what they want. So that's why, yeah, the very last one that what I would like to say is about how the government, even all of the stakeholder, can manage in the better way and the agreement with the local people. Maybe I forget to say something in my presentation because it is related with the access, yeah, the access of the forest. This is still big homework for us, for all of us. This is also about one section on my dissertation, that how the people can manage the access to the forest, because the forest is still. I mean, the forest belongs to the government, but the local people already or have been working there for quite 100 years. You know what I mean? So this
Yes,
is
yes,
about the
yes.
access, basically.
Yeah.
Yeah. So that's what I can say. So we need to re-strategize or revitalize about the regulation in terms of access and land use system management.
Yeah.
Thank you.
Good, sir. Thank you very much. Sir.
Yeah,
Most
I think
welcome.
it's also correlates with the Miss Puspitasari question about the deforestation and also the needs for housing, for example, and then the efforts to preserve the use of traditional plants. Yeah, Miss Astuti, maybe you can give further comments regarding this.
Yeah. Okay. It is from Miss
Puspitasari.
Yeah, Miss Puspitasari. Yeah. Okay, so I try to underline. I mean, like try to re-mention what I already said. Yeah. That's why it's a bit hard for me to say something because this is our problem. I mean, our common problem. This is what I can say. This is something in front of. How can I? In our yard. It's already in our yard, in front of us. So this is the real problem. So what I can what I can suggest in this case is about to find the what is that, like another species that we can also use it for making house, for example. So we can substitute it, and then of course we also can involve. We already have here University of Jember. We already have another university also, so we can collaborate. I mean, between the government, the regional, either Jember government, for example, and regional government, East Java in the level of regional government, and then all of the university here, and then the national park or even the stakeholder which is concerning or which is managing the forest area. So I think all of us can talk together. How could we solve this kind of problem? Because then local community they feel that they used to make housing by using the spaces from the forest. It's common, right, in their tradition. So yeah, the only way that we not the only way, I think. I mean, one of the best solution is just try to find out the substitute for the species, and then the other one, because some species also need another species. I mean, for example, like some herbs need another tree to live inside of the forest. Means that we can also domesticate this kind of species either in the buffer zone area or even in the rural.
Yeah, I think there is a connection problem with Miss Sri Astuti. Yes, I think.
But you can still hear my voice, Miss Afifa?
Yeah,
Yes?
you can.
Yeah, okay. So
I can hear it.
yeah, okay. And the last one is about what did what I said? Domestication. Yeah. We also have a biosphere reserve, Blambangan, right? Correct me if I'm wrong, please. The name Blambangan Biosphere Reserve, as far as I know. And I also intend, maybe I can after I finish my study, then I can help. Inshallah, I can help to develop, and to develop or improve more, or even like how can we just upscale this management into the landscape area. Yeah, inshallah, I will do my best. Yeah, to contribute my my thought, my everything to this kind of. Yeah, this is my my dream also, that I can work in this landscape level, especially in Jember, because this is a long story. I'm the alumna also, or even my study from magister or even PhD. I also do the same thing there. I'm in the same area, so it was like long story, and like it has. I mean, I have a lot of memory about it. So yes, that's what I can say, and I can suggest. Thank you.
Yeah, thank you, Miss Sri Astuti. I think it's also not only responsibility for the researcher, but also government, and also we don't forget it's also responsible for the community itself to conserve our world, and also for in sake of medicinal plants, I think. So if there is any participants who want to ask to our speaker, this afternoon, please raise your hand. Okay, let me see.
Can I ask to Pak Amrun again one more question?
Yes,
Is it okay?
please.
If nobody asking.
Sorry, of course.
Okay. Okay. Pak Amrun, I work in physics, in biophysics. That maybe is sensor is in also in mind of my thing, but difficulty is for the calibration. I think is for yours is easy one for the calibration. The calibration of the to calibrate of the your XR. I mean your measurement or your equipment or your sensor. The process of calibration. Thank you.
Okay. First of all, using if we use to calibrate the scanner, flatbed scanner, we use what we call the color degradation block or color degrading block supported by I don't know, I forgot, by Canon or which one, the other printer, Epson. There are color degradation to calibrate black to white. But if you mean that the calibration is a calibration curve to measure or to calculate something, every purpose has its own calibration substances. For example, if we want to determine total polyphenol content of coffee, which we know that chlorogenic acid is a major polyphenol found in coffee samples, whether green coffee, predominantly in green coffee or the roasted coffee. So we use chlorogenic acid as a standard. We characterize our sensor first before it is applied to coffee samples. So basically, characterization here is validation. So we made a calibration curve that measure linearity response between the chlorogenic acid and the color intensity of sensors. And then we also calculate the detection limit or LOD. We also determine limit of quantification. We also test the selectivity. We also test many things in validation. So after we show that the sensor is okay in the characterization step, then we know that the sensor is ready to apply for a real sample determination. So when we measure the antioxidant capacity of curcuma genus, for example, maybe we can express the antioxidant capacity as a gallic acid equivalent, calculated as gallic acid equivalent. But it is not, I don't recommend, since the most polyphenol found in the curcuma genus, or let's say curcumin or temulawak, which is known as curcuma santoriza, all the curcuma rhizome or curcuma genus, the major polyphenol found is curcuminoids, or let's say the curcumin. So if we calculated the antioxidant capacity, we should calculate it as a curcumin equivalent, which is related or represented what compound, what major compound in that particular extract. I hope it answer your question, Bu Ari.
Yes, thank you very much. It's complicated, but it's complete. I think it's complete seminar to the conference. There is a method. There is the antioxidant. There is people. There is humanity, and there is community. It's very good. I think. Thank you very much for the committee and for the university and everybody, and then Sri Astuti, Pak Amrut that here. It's very good. I think. Okay. Thank you very much.
Okay, thank you very much for your compliments, Bu Ari. And I'm still waiting one or maybe two that who want to ask by live by raising your hand. I don't think so. Maybe I can ask Ms. Sri Astutik for your closing statements, and also Mr. Amrun. Please first from Ms. Sri Astuti.
Okay, thank you so much, Ms. Afifah. Yeah, I think in this occasion, I would like to use this opportunity to say that basically in our life we need to marry between conservation and development, especially in terms of medicinal plants management. So we don't need to spirit both of them, because while we are by using both of concept, it could maintain or it could sustain the medicinal plants for the long run. That's what I can say. And the very last one, all of the stakeholders of the medicinal plants resources should work together and hand in hand, and then especially we need to concern a lot for the community, the local community, because they are the key actor of the development process and development process, or even the conservation aspects. Thank you so much.
Okay, thank you. And so, Mr. Amrun, please give your close statement.
Okay, thank you, Dr. Afifah. I think since the paper-based sensor is developed or fabricated from paper, so it can be easily handled, and it is more safe to the environment, and it is also suited with clean chemistry spirit. Whether we know that in the paper-based sensor, we only use a small amount of regions and or bioregion, and also we also use a small amount or small volume of sample or standard solution. In this case, we already to reduce any chemicals that used, which is it is more less what we call cemaran or contamination to the earth, to our beloved earth. Thank you, Dr. Afifah.
Okay, thank you very much for our both speaker in this afternoon. Very interesting topics. And I'd like to wrap up our plenary session this afternoon. So anything that may be a challenge, maybe it can trigger our future collaboration and our future research. Okay, thank you very much for all participants. So just giving back the time to the moderator.
Thank you so much to Apotheker Afifa Makhlourin, Master of Science, for guiding the last plenary session so well. Thank you once again.
The following agenda is awarding certificates to the speaker and moderator of the plenary session 4. The first certificate is for Ms. Sri Astutik, Master of Science, PhD candidates. Thank you very much for making the time today, ma'am. The second certificate is for Dr. Apotheker Muhammad Amrun Hidayat, Master of Pharmacy.
Thank you very much.
Thank you very much, Doctor.
And the